today? It's gonna be some nice size, 150s.
Yeah, that's so true. But what time is that?
I want to work. Oh, Jesus. Wait, what time does he want to go?
He just says afterwards, so tonight.
Okay, guys, you ready to go? Okay, good. Video stop working. Okay, we are recording. Patrick, can you hear me? Here he is. We're in.
Yep, I can hear you.
Good, I can hear you too. So some people have not taken the exam yet. That's okay. We're going to move on today. We're going to do chapter 11. Hopefully I won't finish it, but we'll see. Chapter 11's kind of more in depth on apoptosis. So we already kind of did the broad brush, but we're going to go into more detail on how the caspases get activated, how the BCL2 proteins work. What else? And then once the cell goes into apoptosis and becomes apoptotic bodies, we said macrophages come and take them out. It's actually a more complicated process. The apoptotic bodies are calling the macrophages towards them and then telling the macrophages that they've hit the right target. So we'll talk about those molecular aspects of that. What else? I guess that's about what we're going to talk about today. What's the other thing? Oh, autophagy. Hopefully we won't get that far, but we'll see. Some really complicated stuff, so let's hope we can enjoy it as we go. Another thing too is okay. Another thing is I'm going to try and bring in some of the old chapters as we go through here too, because like I say, it's hard to do anything in cell biology because it's all happening at once. So some of the things we talked about earlier are going to come into play here, and I'll hopefully review some of that for you. So anyway, here we are, apoptosis and autophagy. So apoptosis we've talked about is basically just death, and autophagy literally means self-eating. So we'll talk about why that happens and how that happens later in the chapter. But basically, it's like it's a starvation response of cells and of whole individuals when they're at low nutrients, and it's how to preserve life over time when their nutrient levels have dropped dramatically. So hopefully we'll get to that Wednesday. But anyway, we've already talked about your extrinsic pathway and the intrinsic pathway of apoptosis. Today we'll see how the extrinsic pathway also activates the intrinsic pathway, which I didn't mention last time, but we'll get into that today. And we'll talk about how this actually happens, what's causing these things to get together. So we'll start out there with our caspases. So your procaspase, remember I said earlier that it was activated by proteolysis. We've got a prodomain here that's going to block the proteolysis. But we've got also a large subunit and a small subunit. And what happens when you get these together is, again, like I keep harping about getting proteins together, because when you do, then the enzymes are near their substrate. So when you get several of these together, they cleave in two different parts. So the one cleavage part takes off this prodomain, which is inhibiting the protease. And then the other is part of this small subunit. So it cleaves here and here, and then it forms this, I guess you would call it homodimer or heterotetramer. I'm not really sure. I think homodimer because the large and small are from the same protein. But once they're cleaved apart, it kind of looks like a heterotetramer. But anyway, you've got your regulatory subunits on the inside, small ones, and then the catalytic on the outside. What else do I want to say? Oh, also, these caspases, though, each caspase is going to have a specific target sequence it's looking for. So just like kinases, I know you probably talked about kinases in biochem. Kinase isn't just phosphorylating every serine or threonine it finds. It's looking for a consensus sequence around that serine or threonine to make it more specific. And these caspases are similar in that, like, for example, caspase 3 is not just going for your aspartate, but aspartate, glutamate, valine, aspartate. That's the sequence it's looking for. And then it'll cleave after that fourth aspartate there. So there is a lot of substrate specificity here. And I was joking the other day, a couple weeks ago, about how, we only talked about caspases 3, 8, and 9. But obviously, if there's a 3, 8, and 9, there's a lot of other ones too, because they're numbered based on the order that they're found. And we didn't talk about the rest of them because they weren't important for apoptosis. Here you see caspases 1, 4, 5, 11, and 12 are all involved in the inflammation pathway. Here are our initiator caspases, and then our executioner caspases, and then 14 is only involved in keratinocyte differentiation.
Are caspases involved in like other pathway other than apoptosis?
Yeah. So the question is caspases involved in pathways other than apoptosis? So these top 5 here you can see are only involved in inflammation. These middle ones are all apoptosis, and this last one is keratinocyte differentiation.
In terms of inflammation, do they work in reducing it or causing it?
Yeah, I don't know the inflammation stuff that well. Yeah, I'm not a big immunology. Again, you're always here finding a hole in my knowledge. Yeah, I don't know much about immunology, so I can't really tell you about that. But anyway, let's focus on the ones that we do care about here. Our initiators and our executioners. So each of these has got a prodomain, which is this part out here. And that's, that initial part that I said gets cut off. And then they've got the large and small subunits, which will then be cleaved apart. So this prodomain is going to help, in addition to like blocking the activity, it's going to help them to accumulate together so that they can activate each other. So we've got 2 different types of these association domains. One is the extrinsic pathway, which is the death effector domain, DED. And then the intrinsic pathways are the CRD or the, caspase recruitment domain. These are all the extrinsic pathway again, and these are the intrinsic pathway. And those are your initiators. So you can see the initiators are going to clump together. Once they get activated, they're going to cleave off the prodomain of the executioners, and they become active. So we don't have these domains in the executioner caspases. And also, if you remember we'll see in the next slide, but FADD also has this DED domain, and the APF1 being part of the intrinsic pathway has the CARD domain. And basically your DED domains will bind to other proteins with DED domains and CARD the same way. And that's what we call this homotypic binding to each other. They're the same domains, and they'll bind to each other. So here, again, a little more detail on how these pathways get activated. If you remember, the TNF receptor is a single peptide. It's got an external domain, single transmembrane domain, and then this internal death domain. And it will be swimming around in that membrane until the TNF alpha comes along and binds to one of them, and then the other 2 will come in and bind to that TNF alpha as well and form a trimer. So again, in recruitment, once you've got that trimer together, the death domains are binding to each other, that brings up FADD. And you can see why it's called an adapter protein. You've got the death domain on one side and the death, I always forget this, death effector domain on the other side. So the DD on one side, the DED on the other side. And now The FADD then is going to associate with the caspase 8 through this DED domain. And that's going to cause the caspases to be close to each other and then start to cleave each other. And that's how we get activation of caspase 8. Oh, question.
So all the caspase like attachment, is it happening on the cytosol or outside exosome?
This is all in the cytosol. If you look here, this is your plasma membrane. Here, let me just blow this up. So here's your plasma membrane. This is extracellular. That's what TNF alpha will bind, and then your intracellular domains are all involved in this oligomerization to get the caspases together. And then, once that caspase is activated, it will activate the caspase 3. So here is our apoptosome already put together. Basically, here's your APAF1. Again, it's a adaptive protein. It's got the CARD domain on one end and this APAF1 domain on the other. So this domain over here is what's going to bind the cytochrome C. So this is folded up when it's not activated. It's folded up so that APF1, this green domain here is hiding the CARD domain. And then cytochrome C will bind to this green domain out here, and that will cause it to open, and then ATP binds to it, and that opens more. And then you've exposed this CARD domain. And once the CARD domain is exposed, it binds to caspase 9, and then that starts the whole process of assembling the apoptosome. And once it all gets assembled, these caspase 9s are next to each other, and they activate each other. Again, just kind of cleaving here and here, getting rid of the prodomain, and then reorganizing to form this homotetramer and homodimer. Okay, is that good? Okay, so mainly, the only thing we're really adding here is how these oligomerizations are happening through these domains. And then your executioners. So you cleave off this pro domain out here, and then they'll cleave again over here and then form that executioner structure. And then here we've got several targets of these executioner caspases. One is PARP, poly ADP ribose polymerase, and this is an enzyme that's really important in DNA repair and maintenance. So this gets inactivated. So once the DNA starts getting broken down, it's not going to be available. And then we've got gelsolin. Remember gelsolin from chapter 3? Can I bring that up? Let's bring it up. We got time here. Okay. So gelsolin, we're going to talk about gelsolin. Here's one example of gelsolin. So in the back here, gelsolin is cleaving the microfilaments. So let's see, I can't find that. God, I've seen these slides my whole life. It's cross-linking. Yeah, I'm not seeing it now. But at the time, we were talking about how when you polymerize new microfilaments, basically you're breaking down in one place, moving the monomers, and then re-polymerizing somewhere else. So gelsolin is involved in cutting in the middle of a microfilament strand so that you've got now 2 plus or minus ends, because the minus ends are where you're going to get depolymerization. So getting back to chapter 11, activate gelsolin, and that's going to cause breakdown of the microfilaments. You're also directly breaking down actin, again, breaking down microfilaments. And again, I repeat myself a lot. I think it feels like the more you hear it, the more likely you're going to remember it. But If you remember, I said that in apoptosis, when you start breaking down the cell cortex, which is all actin microfilaments, you start losing integrity of the plasma membrane, and that's where you start getting those blebs. And then vimentin is another chapter 3 protein that's involved in the focal adhesions. Well, that's penicillin actually. But vimentin is another actin binding protein that will then cross-link it to other proteins. So vimentin also gets broken down, which is again cytoskeletal breakdown. Our third targets are ICADs, inhibitors of caspases. And we haven't talked about these yet. We'll talk about them shortly, so I'll get into them now. And they're also going to break down signaling molecules. Again, when we talked about cell division, I said whenever you activate cell division, you have to inhibit apoptosis, because if you overstimulate the cell to divide, it can then go into apoptosis. So you have to, there's always a balance between cell division and apoptosis. So we were talking back then about AKT, and I'll talk about it again later today. And AKT is phosphorylating bad, and BCL2 protein, and FOXO, which is a transcription factor that then goes on to upregulate apoptotic proteins. So we're going to block the pro-apoptotic BAD and block the transcription factor that's going to prime the cell more for apoptosis. Our MAP kinase is also going to inhibit apoptosis. It's more later. And then inhibitor of caspase activated DNase. So again, back in chapter 10, when I was talking about apoptosis, I said you get activation of DNases that break down the DNA. You get those 200 base pair pieces because this DNase is just randomly cleaving between nucleosomes and breaking the DNA down. Well, before you activate apoptosis, you've got this protein there, inhibitor of caspase activated DNase, ICAD. So ICAD is inhibiting that DNase. When apoptosis gets activated, the caspase then breaks that down and the DNase is activated. So we've got inhibition of DNA repair, breakdown of the cytoskeleton, including the organelles, activating or inhibiting the inhibitors of caspases, and inhibiting the ability of signaling pathways to block apoptosis. So we're pushing apoptosis forward. And here's our IAPs. So again, IAPs are always there in the cytosol, just like you always have procaspases around. They're always there. And they're waiting for the opportunity to block caspase once it gets activated, caspase 3 here. And this is going to be regulated very similarly to it's regulated through that intrinsic pathway in the same kind of way. So basically, BCL2 is blocking release of cytochrome C, and it's also blocking the release of this other protein called SMAC Diablo. So SMAC and Diablo are basically two different names for the same protein. Again, two different people found it in different species or whatever, so it's got two different names, but it's the same thing. So SMAC is going to inhibit the IAPs. So here we are, BCL2 is holding back the flood here, keeping cytochrome C back and keeping SMAC back. And then when something like a pro-apoptotic BCL2 like Puma comes along, cytochrome C can be released, causing apoptosome formation, and also SMAC is released, which is going to inhibit those inhibitors of apoptosis, the caspase inhibitors. Okay, so just backing up again. That's our inhibitors of caspases, ICADs, and we're inhibiting them here. Actually, this shouldn't be IAPs, it should be ICADs, but I think they're called both. Anyway, so that's our SMAC Diablo pathway. Okay, so now we'll talk more about this. What's this BCL2 process going on? And again, we're talking about association domains, domains that cause proteins to associate with each other. And so we've got three different groups of BCL2 proteins. Group 1 is our BCL2, BCLXL. So these are our anti-apoptotic proteins. And again, BCL2 being the first one that was found. So we've got 4 domains that show homology across BCL2s, plus a transmembrane domain. So because BCL2 is the first one found, we call it BCL2 homology domain 4, 3, 1, and 2. So we've got these homology domains that are going to cause this homodimerization later. And mainly it's this BCL3 domain that's going to cause these proteins to clump together. Keep in the back of your mind too this transmembrane domain, because it's going to be involved in making a channel in that mitochondrion so that we can release cytochrome C and SMAC Diablo. So that's our apoptotic with multi-domains. They all have multi-domains. Then you've got 2 different types of pro-apoptotic BCL2s, some with the multi-domains and some with only the BH3 domain. And you really want to think about these 2 down here as being kind of like initiators. So these are going to activate the back and backs type of BCL2 protein. Let me make sure I didn't forget something here. So bad. Okay, so regulation here. BID and BAD are the ones that are going to initiate. So these are regulated. If you remember back, get to it here. Let me just talk about it now. If you remember back to here, this was chapter 9 when we were talking about the AKT pathway. So receptor activation, activating PI3 kinase, making PIP3, activating PDK1, and mTOR-RIPTOR, and they phosphorylate AKT. And AKT then activates the protein synthesis pathway, increases metabolism. Notice this guy here, mTOR-RAPTOR. He'll come up later today too. So anyway, but again, when you're activating cell division, you have to inhibit apoptosis. So AKT is phosphorylating BAD, and that inhibits BAD. And we're phosphorylating FOXO, which inhibits FOXO. FOXO would normally upregulate BIM and FAST ligand. And then also, inhibiting cell cycle over here. And also, AKT is going to increase the amount of MDM2 around. And MDM2 is decreasing P53, so that's inhibiting apoptosis too. And another one that isn't on here is CREB. CREB is a classic transcription factor that turns a lot of things on. CREB pro-growth is going to activate the transcription of BCL2, so that's inhibiting apoptosis. So just review again. AKT is phosphorylating BAD and BIM, which are our BH3 only proteins, and these are going to cause sequestration of BAD by this protein called 14-3-3. So it's a series of proteins, I don't know if you can see that, called 14-3-3 proteins, which in my opinion is the #1 protein in cells that needs a new name. 14-3-3. It's basically a scaffolding protein. It holds on to other proteins, and its function is like if the cell wants to turn a protein off, it has it bind to the 14-3-3, and it sits there and waits until it's ready to be activated again. So when AKT phosphorylates bad, it gets bound to this 14-3-3, and then it's no longer available to activate apoptosis. We're phosphorylating FOXO, which is causing downregulation of pro-apoptotic proteins, BIM and Fas ligand, activating CREB, which isn't up here, increasing BCL2, and then increasing MDM2, or activating MDM2. I think what this has to do with this phosphorylation, again, remembering back to chapter 10, when AKT phosphorylates MDM2, which is being translated in the cytosol, it causes translocation into the nucleus where it can act on P53. See if I can find that real quick, just so you know I'm not lying. Where's that chapter? Oh, that was an exam 3. Again, I like to try and get across a sense of the gestalt of the whole thing, that these aren't individual things happening. Everything's happening together here. All right. Here's another trick I'm going to let you in on. I often use masks in my slides. Hi there. Yeah, I'm not going to find it here. Anyway, back to chapter 11. So anyway, these are all the things that AKT are doing to inhibit apoptosis. MAP kinase, on the other hand, is phosphorylating again bad and BIM. BIM is going to be activated by caspase 8, we'll see in a minute. But BIM is being ubiquitinated and degraded. Bad is being phosphorylated and binding up in the 14-3-3. It's also phosphorylating caspase 9, inhibiting activation, and activating CREB again, increasing transcription of BCL2. So again, just repeating myself over and over again, here we are, pro-growth signaling pathways are inhibiting apoptosis through these mechanisms. All right, so backing up, get back to the BIM story. Okay, so this is kind of interesting. Again, we're talking about this BH3 domain binding to other BH3 domain proteins. We've got a BH3 domain protein in BCL2, and then we've also got BID, BAD, BAX, which have got BH3 domains as well. So what happens after we get this death domain activation over here, TNF receptor or FAS here, activating caspase 8. Caspase 8 then activates caspase 3, which we said before. But another thing caspase 8 is doing is to cut that pro-domain off of BID. And now BID is one of those single domain activator BCL2s. So it's going to bind to BCL2 and block its activity. So BCL2 can no longer bind to BAC or BAX when it gets inserted. And it also activates BAC and BAX, which are then going to bind to each other and form this pore in the mitochondria. Excuse me. So that's how we get activation of or release of cytochrome C and SMAC Diablo, because we've got our TET4 domain BAC and BAX is coming together to form this channel here, and TBID is causing that to happen. Any questions about that? Am I going too fast? No?
I think like after a review, like on will be able to like,
Okay,
this is you're doing good.
okay, so that's our BCL2 proteins. All right, so that's the whole thing in terms of what we've added to apoptosis, which is how our caspases getting together and activating, and how are the BCL2 proteins coming together and activating. It all has to do with these homotypic domain binding to each other. Okay, so that's it for apoptosis. I promise I won't talk about apoptosis again, but you know how I lie. Okay, so here we are, clearing out apoptotic bodies. So again, the cell gets rid of an apoptosis cell really, really fast. And it's kind of important because there are a number of diseases that happen when you don't get rid of those. The apoptotic bodies are going to activate an inflammation response. And we all know inflammation is bad, right? Short term fixes things, long term causes a lot of problems. System lupus erythematosus is chronic systemic inflammation, and the lymph nodes get clogged up with uncleared apoptotic bodies. CPOD, you guys know what that is? Did you guys have grandfathers who were coal miners like me? Grew up coughing and hacking. CPOD, and my mother was a smoker. She had CPOD. So anyway, when you've got a lot of cells that are going into apoptosis, these apoptotic bodies build up, and that causes even more problems. So anyway, you need to get rid of those things fast. And we're going to get rid of them by both dedicated phagocytes, like your macrophages and dendritic cells, things that are always there looking for something to eat, and then your induced phagocytes. So endothelial cells, epithelial cells, and fibroblasts can also do this phagocytosis when they get induced to do it. So how does this happen? So no, that's not it. Find these signals. So again, I'm going to go back to chapter 2. Remember us talking about our phospholipids. In this particular case, what are we looking at? Phosphatidylcholine. So phosphatidylcholine is this guy. Okay. So phosphatidylcholine is like the highest constituent phospholipid in the membrane. And if you remember, phospholipase C will cut it here, and phospholipase A2 will cut here. So phospholipase A2, when it cuts this off, leaves your glycerol with one fatty acid and the choline attached. And that's called lysophosphatidylcholine. Lysophosphatidylcholine, which is here, LPC. So that gets released. And then we also get phosphorylation of sphingomyelin by this enzyme sphingomyelin kinase, and that gets released. And this protein, factokine, so it's a cytokine that's specific for this process. And then nucleotides. So we see this a lot with, I was talking about it earlier with conversation between neurons and glial cells, where nucleotides can be released and cause signal transduction. Here we've got nucleotides being released from an apoptotic cell. So all these then act as chemotactic attractors. So that's why they call them find me signals. So when the cell apoptosis starts releasing these signals, and any macrophages in the area will sense that. We're binding here to, looks like 7 transmembrane receptors. This is the adenosine receptor here, purinergic receptor, and these other receptors. So anyway, that then activates cell crawling. No, these are phagocytes, so it would be amoeboid movement. and they're moving towards the apoptotic bodies. Did I forget anything here? I'm a little out of it today. I just came back from Pennsylvania. An aunt of mine died last week, and I just got back around 10 o'clock last night. Got to sleep about 12:30, so I'm a little out of it today. Okay, so that's the find me signals. You're releasing all these chemicals, and that's causing phagocytes to move. Once the phagocytes get there, the apoptotic cells have eat me signals. So one of them, a really important one, is this phosphatidylserine. And again, we talked about this way back in chapter 2. We talked about this. Remember this from chapter 2 with our flippase and floppase and scramblase. So your normal membrane of cells is the outer monolayer. It's going to be mostly phosphatidylcholine, also sphingomyelin, a little phosphatidylethanolamine, and all the phosphatidylserine is on the inside because flippase is constantly putting it back on the inside. Keep PE in mind too, because PE is going to come into play when we get into autophagy. Okay, so normally flippase is putting phosphatidylserine on the inside, and then when it gets turned off and scramblase gets turned on, it evens everything out. Right? Scramblase is going in both directions, so everything gets equal, which means now you've got phosphatidylserine on the outside of the membrane. So that's chapter 2. Now here we're going to see that exposed phosphatidylserine then happens here because of caspases. Caspases will cleave the flippase and inhibit it, and cleave the scramblase and activate it. So that causes that PS to be exposed. And then you've got receptors on macrophages for phosphatidylserine. Isn't it amazing how everything has to do with specific receptor interactions? Wouldn't it be great to be a pharmacologist and you could study this all day long? Or maybe you could be a doctor and save lives. One or the other. Okay, so you've got these PS receptors here. You also have some bridging molecules that go between PS and the PS receptors. But basically, this is causing the macrophage to grab it. And also, once you activate this, once you activate these receptors on the membrane, that activates our old friend RAC. Remember RAC? RAC-RO CDC42, right? These are all the G proteins that are involved in rearranging cytoskeleton. So now you activate RAC, and cytoskeleton starts to rearrange and it forms a cup. And the cup kind of, as it binds to receptors on that enzyme, it kind of forms itself around that big chunk of apoptotic cell. I'm not crazy about this drawing because your macrophage is smaller than the apoptotic cell. Your macrophage should be like huge compared to it. But anyway, we're gonna like, how could you engulf this in that? Question?
Yeah. What's the function again of the bridging molecule?
I'm not sure exactly. It's doing the same thing as if the PS is binding directly to the PS receptor. So I'm not really sure why there are bridging molecules, but there are.
In terms of find me or eat me signal, aren't TNF alpha involved in either of those?
No, this is downstream from that. Okay, so TNF alpha activating the extrinsic pathway, caspase 8, caspase 3, and then caspase 3 is going to do this. Okay, and that's going to expose the phosphatidylserine. And I imagine, I didn't read about this in the book, but I imagine it's causing this to happen too. Probably also activating the find me signals. Okay, so a phagocyte has found it. And once it does find it, it engulfs the whole thing. And then from there, you get into chapter 3 with ATPases. If you remember then, back then we had 4 different types of ATPases. There was the P type, and the second type was the V type, which are on vesicles. So here, you're going to have these small vesicles called, oh God, I can see the figure in my head. That's the problem with being a visual learner. The words don't come sometimes. Oh, I can't remember, but small vesicles that have these ATPases in their membrane are going to fuse with that phagocytotic thing, and they're then going to acidify that phagosome, and so that's going to start to break things down, and then those phagosomes will then lyse with the lysosome, releasing all your acid hydrolyses. They're going to break down all the macromolecules in that chunk and release the monomers, and then the cell will then just use those monomers for cell growth. And then this is interesting. Once the phagocyte has phagocytosed that the apoptotic cell starts to release anti-inflammatory molecules. So now we're going to start to inhibit inflammation. So once the problem is gone, you want to stop inflammation. So that's going to be part of the mechanism as well. Okay. That's all I have here. Any questions about this? Basically, find me and eat me. Find me releasing small molecules for chemotaxis, and eat me, sticking out as something that needs to be phagocytosed. Okay, that's 40 minutes. Shall we go on? I hate to steal my thunder. I only have a few slides on autophagy, but we keep going through material really fast. See, in my undergrad classes, I'm writing notes on the board, and this would have taken me an hour and 15 minutes to do, but here we are, 40 minutes in, and I'm already done with it. But I don't know. We good for today, or should we keep going? Is that enough? That was a lot, and you can see why I didn't want to add this onto the last test. I mean, all this in addition to all of that other stuff.
In terms of like a process of macrophage or like heating it up, does it everything gets degraded or some of the stuff gets recycled?
Yeah. So the question is, when after you form this phagosome and bind with the lysosome, does everything get degraded? Yes, everything that should be degraded gets degraded. Now here's why I'm qualifying that. Getting back to my grandfather, the coal miner, okay, with CPOD, what happens in something like that, or in like asbestos or something, silicon dioxide, get chunks in your lungs, phagocytotic cells will surround them and engulf them, can't break it down because it's rock, right? And then those cells will die and they'll become apoptotic. And other macrophages will come in and eat those apoptotic pieces, and they'll die because they can't digest silicon either, silicon dioxide either. So as long as it's a normal, healthy pathway, yes, everything gets degraded and released as monomers. But if it's like in your lungs and you're getting chunks of stuff in there, that begins a disease process. I'm keep bringing up my grandfather because I was just in my hometown yesterday. So it's forefront in my mind. Okay, is that it? All right, warning, this is pretty complicated. Pretty cool though, I love a cool pathway. So let's call it, let's stop for today. And on Wednesday, hopefully I'll have your grades. I won't have your exams graded yet. Maybe I'll do as many as I can. Some people need another day, so they're taking it tomorrow. So I may or may not be able to go over the exam on Wednesday. We'll see. This afternoon, I'm going to start grading the ones that are already done. You guys finished already? No? Okay. Did I get an email from you this morning?
No, that was last week.
Oh, okay. All right, then I guess that's all we got. Matt and Pat, we okay?
Yep, we're good. Sorry about the loss of your aunt.
Oh, thanks. Yeah, it was sudden. She was going into dementia, and It's getting worse and worse. And she slipped and fell down the stairs backwards and got a blood brain bleed. Kind of I don't know, pretty horrible, but yeah. Anyway, all right. So we'll see you again on Wednesday, and have a great